So the motivation for our first company, Seia Bio, was nothing to do with what the company looks like now. So I figured I'd just talk a little bit about the pathway to how Seia got to where it is. So what we were originally thinking about in my group is how we can study microbial biodiversity. So along with climate change, loss of biodiversity is one of the major negative impacts on our globe right now. And as we can see, we've lost a significant percentage of the global number of species. These are basically the number of species that have gone extinct since 1970. And importantly, microbes really are the basis for all life on Earth. They cycle nutrients in soil. They help protect both plants and animals and humans from external pathogens. And they help us metabolize our food in our guts. And so as we lose that biodiversity, we become more susceptible to disease, our soil becomes less healthy, and we are less able to sustain our global population. So we wanted to take microbes from the environment and study them in our lab. The problem is, soil is a nice, cozy environment for the microbes. When we take a sample of microbes from the soil and try to transport them into the harsh environment of a lab, we lose about 90% of the species. And so our goal was to figure out a way to just take microbes from their natural habitats and bring them to our lab. What we decided to try was a self-assembling material that forms on the outside of individual microbes and kind of acts like a suit of armor. You can also think of it like the coating on the outside of the M&M that helps prevent the insides from melting. So what are these suits of armor made of? Well, we wanted to think about things that were inexpensive and easy to access. And if we were going to be using them on microbial samples that were coming from things like soil, we also wanted them to be food grade. So we found materials that were derived from polyphenols, which are things like green tea extract or tannins from coffee and tea, as well as vitamins and minerals that you take every day, things like iron and manganese that are found in supplements. And when you mix these two things together, they self-assemble into this very thin coating that forms on the outside of microbes. And these are called metal phenolic networks because they're made of polyphenols and metals. And I'll be referring to them as MPNs. And these are really cool because they form really easily just by mixing these components. So if you watch this video here, this is just the microbes being coated. All we have to do is mix the metal with polyphenols and microbes, and that blue color is the coating forming. And as we change the components of these coatings, we change the physical properties of those coatings. And that's what gives us these different colors shown here. And if we image microbes after they've been coated, we can see that these coatings form homogeneously on the surface. And we think that this is really important for actually protecting the microbes. So this is just an uncoated microbe. And this is a microbe that's been coated with a layer of those metal phenolic networks. And by TEM imaging, we can see that the metal ions here are what's giving us the kind of sharp contrast. And that microbe is fully coated. And because these coatings are very thin, they're about 2 to 10 nanometers thick, we can actually coat these subcellular structures. So this really allows us to protect the whole microbe. And what we see is that when we coat these in batch, so obviously we're not going to do this one microbe at a time, we get uniform coatings across all of the microbes. So this is just a stain that we've put on the coating, and you can see here the shell structure that forms. And each of the microbes in this matrix is completely coated, and they're all evenly coated. And what we found is that these coatings protect against pretty much every stressor we could think of throwing at them. This ranges from things like oxidative stress, putting them in hydrogen peroxide, UV and radiation exposure, temperature fluctuations, chemical exposure, mechanical stressors, and high salinity. And so we thought, well, maybe this could be useful for things beyond just preserving microbes to bring them back to the lab, right? Maybe we could actually use this to improve human health. How many people in here take probiotics regularly? Awesome. About 99.9% of the microbes in that are dead. And that's because there are a few species that we know how to protect really well, but the manufacturing process is very harsh on them. And the problem is probiotics companies are basically marketing companies. And so they don't care how many live microbes they give you. They just want you to buy their product. And so we know how to manufacture these. We get about 0.001% viable cells. But there are many microbes that we think could be beneficial for treating things like Crohn's disease, IBS, and other kind of diseases that would require an FDA-approved treatment, but we can't actually manufacture these because they're very delicate. So what we thought initially for our metaphenolic networks was that we could actually use them to help us move beyond the probiotics you buy at the grocery store.